Okay, welcome, my dear friends. Today's our eminent resource person, Dr. A.SHAILAJA Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Autonomous, Siddipet. I'm going to explain one of the most important topic in BRYOPHYTA, that is ANTHOCEROS, Anthoceratopsida type study. So, Madam, please welcome and start your session, Madam. Please welcome.
Thank you. Today's our topic is Anthoceratopsida. It's very, very important. My voice is clear?
Yes, ma'am.
Okay. Shall we start the class? Good afternoon, my dear students.
Yes, ma'am. You can start class.
Okay. Anthoceratopsida. First, I will tell you that general characters of Anthoceratopsida. This is our second class. First is the hepatic apsida is completed in the yesterday's class. Today we are going to discuss that Anthoceratopsida. Same like that, the sporophytes of Anthoceratopsida plants are similar to long blades or horns. So that's why these the group of these Anthoceratopsida members are called hornworts. Okay, hornworts. This is the image of hornwort. Okay, so in this class, first classification, introduction, habitat and habit, thallus structure, internal structure of thallus. After that, reproduction, sexual reproduction. In that sexual reproduction, first male and female sexual organs, antheridium and archegonium developments. After the fertilization, and after that, sporophyte structure. Okay, this is the type study of Anthoceros. So, first, what are the special features of Anthoceratopsida? These are called hornworts. Okay, remember that. And gametophytic structure, we are coming to the gametophytic structure. Same, these are dorsiventrally differentiated, and dorsal surface is green in color, ventral surface is colorless. Same like that hepatic apsida. Okay. So at the ventral side, we are observed that smooth type of rhizoids, which helps in absorption and fixation. In the internal structure of thallus, mucilaginous cavities are present, blue-green algal forms that nostoc and anabina live in symbiotic association in these cavities. That is the specialization of Anthoceratopsida. Okay, remember that. What is the special character, Nana? In the structure of the thallus, Anthoceratopsida general characters cheptunnan nen first. Okay, na? Mucilaginous cavities are present. That cavities having the blue-green algal forms. That example for that nostoc and anabina. These contact with live in symbiotic association in these cavities. Okay, this is the special characters of Anthoceratopsida. Chloroplasts contain naked pyrannids, which is an algal character. Okay, so that in this Anthoceratopsida members having the chloroplasts that contain naked pyrannids. This is a which is an algal character. And sex organs in Anthoceratopsida are embedded in the thallus. Male sexual organ is called antheridium and female sexual organ is called archegonium. Sporophyte consists of foot and capsule. Seta is absent. So in these members, we are observed in the hepatic apsida members of the, for example, in the Markantia, already we seen that in the Markantia sporophyte, the three parts are there: foot, seta, and capsule. Okay, so
Ma'am.
Naina, tell me.
Ma'am, now what is your explaining it? The screen is not available, ma'am.
Just general characters, ma'am. General characters of Anthoceratopsida. Okay, so in this classification,
screen I can't see now.
Yeah, classification is there in the scene.
Classification is there, but general class is not coming, ma'am.
No, ma'am. This already we are discussed in the last class, na. So just
okay, ma'am. Okay, ma'am. Okay.
Okay. So that sexual organs are Anthoceratopsida. Those are embedded in the thallus. Male sexual organ is called antheridium, and female sexual organ is called archegonium. Okay. Sporophyte consists of foot and capsule. Seta is absent. So that is the special character of Anthoceratopsida. In this capsule contains spores and pseudo-elaters. In the Markantia, we are observed in the capsule elaters. Okay, hygroscopic in nature, diploid cells, and observe the water and push the spores to outside from the capsule. That is the elator's function. Okay, so same like that. In the anthoceros, we are observed that in the capsule pseudo-elaters. Okay, remember this. These are the very important topics for the entrance. Okay, elaters, pseudo-elaters. What is the special character? In this Anthoceratopsida, that mucilaginous cavities are there in the thallus. That is the symbiotic association with the blue-green algal forms. Okay, this is the special character. What are the special character? You remember. Okay. In this again, next one is the middle of the capsule is columella, which occupies major part of the capsule. That capsule wall is chlorophyllous and contains stomata, helping in gaseous diffusion. Sporophyte of Anthoceratopsida group is partially dependent on its gametophyte, but completely dependent of Markantia sporophyte. But in the Anthoceratopsida, this is partially dependent on its gametophyte. That is the difference between Markantia and Anthoceras. Okay, so now we are discussing that Anthoceras classification. See here, division Bryophyta, class Anthoceratopsida, order Anthoceratates, family Anthocerataceae, genus Anthoceras. Okay, this is the classification of Anthoceras. This is a one type study of Anthoceras. Introduction, habitat and habit. It is commonly known as horned liverwort. Okay, hornwort. Actually, hornwort. Anthoceras represented with about 200 species and distributed all over the world, growing in moist areas. Totally 200 species are there, distributed all over the world. But in that 200 species, 25 species are reported in our India. Common species among them are Anthoceras chambensis and erectus and crispus. Okay, so next one is that Anthoceras himalayensis and candalensis and langti, etc. These are Anthoceras species. Kashyap reported from Mussoorie, Kulu Manali and Chambal Valley, Punjab, Tamil Nadu and Travancore. These three species are reported by the Kashyap from these places, Mussoorie and Kulu Manali and Chambal Valley, Punjab, Tamil Nadu. Okay, that next one is the Anthoceras Shivanandini grow in the hills of Kerala and Anthoceras crispulus from Araku and Andhra Pradesh. Okay, Vishakhapatnam and Uma Maheshwaram hills of Telangana at Mahbub Nagar. So these species are observed in the all over India. So next coming to the thallus structure. This is the Anthoceras image. So see here, this is also gametophyte, is dorsiventrally differentiated. Dorsal surface is green in color due to the presence of chlorophyll and the photosynthetic region of the thallus. Already same points discussed in the Markantia. So that's why I'm going to just speed. Okay, na? If you have any doubt, you'll ask me. Okay, na? Type study complete కావాలి కాబట్టి just fast గా చెప్తున్నాను అన్న, ఏదైనా డౌట్ ఉంటే అడగండి. ఓకేనా, క్లియర్? So next, ventral side. This ventral surface is colorless due to the absence of chlorophyll. So that smooth rhizoids arise from this side and these which is helps in the absorption and fixation. Rough rhizoids and scales are absent in the Anthoceratopsida. Remember this. This is a very important. Rough rhizoids and scales are present in the Markantia but absent in the Anthoceras. Okay, what are the special characters you have observed? Naina.
Ma'am.
Okay, na clear? Jayant Reddy, Maunika.
Ma'am, clear, ma'am.
Okay. So next, thallus exhibits irregular dichotomous branching with insignificant midrib. Okay. So a few small rounded dark bluish green thickened spots are visible. These are the mucilaginous cavities. So about the mucilaginous cavities, I will explain in the thallus structure. Okay, internal structure. In this cavities, colonies of blue-green algae, nostoc and anabina are present. nostoc inhabits of the thallus. Generally, the gametophyte bears long green horn-like sporophytes. Due to this, the plant are called that horn plants. Okay, hornworts. See here. So in this plant, Anthoceros, these are the horn-like structures. Okay, sporophytes are horn-like structures. That's why we are called hornworts. Anthoceros may be annual or perennial. Okay, so this is the Anthoceros annual example is erectus and a perennial example is Himalayensis. So next one, we are coming to the internal thallus of Anthoceros. This is the internal structure. So I ask some questions from Markantia. What is the difference in the internal structure of Markantia and Anthoceros? What are the parts we are observed in the Markantia? Tell me anyone. In the internal structure of Markantia, what we observed? Jayant Reddy, Maunika, Naina, tell me anyone. Are you note down yesterday?
Yes, ma'am.
What are the parts in the Markantia thallus structure? You don't know? Forgot the
Ma'am,
Markantia.
I don't know, ma'am.
Epidermis, assimilatory zone. What are the air chambers? You asked now, yesterday in the yesterday's class. Assimilatory zone, epidermis, assimilatory zone, and storage region. The three parts we are observed in the Markantia thallus.
Ma'am, what, ma'am? Epidermis?
Epidermis, assimilatory zone. In that chlorophyllous filaments and air chambers are present in the assimilatory zone. And the next storage region. In the storage region,
Okay,
are filled with that parenchyma cells.
Okay, ma'am.
గుర్తొచ్చిందా ఇప్పుడు?
Yes, ma'am.
So same like that internal structure of thallus of Anthoceros, we are observed this internal structure, upper epidermis and lower epidermis. But see here, smooth rhizoids only present at the lower epidermis. So in the Markantia, that rough rhizoids and the scales are present. Okay, smooth and rough, two types of rhizoids are there and the scales also there. But in the Markantia only, in the Anthoceros, only smooth type of rhizoids are there. Okay, upper epidermis, lower epidermis, middle parenchymatous tissue. See here,
ma'am.
In this diagram, Chudanana, in this diagram, in the middle parenchymatous tissue, that algal colonies, this is the mucilage cavity, algal colonies are there. Okay, so at the center of middle parenchymatous tissue, we are observed that mucilaginous cavities filled with the algal colonies, that is nostoc and anabina. So that is a symbiotic association. Okay, are you understood? See here, undifferentiated thin-walled polygonal cells of parenchymatous tissue without intercellular space is filled between these layers. Okay, this is the upper epidermis, this is the lower epidermis. All the cells in the thallus are similar in nature. Chloroplast in lamellate type with a marked pyrenoid. See here, this is a pyrenoid is there in the chloroplast. That is algal character. So this is assumed algal character of Anthoceros, which is considered a primitive feature. Okay, that is primitive feature. What is the primitive feature of Anthoceros? Remember that is the what is a pyrenoid in the chloroplast is a rare character in bryophytes. This is a special character. Note down. Okay, this is the algal character of Anthoceros, which is considered a primitive feature. Okay, the dorsal side of the thallus presents some mucilage filled cavities. In these cavities inhabit blue-green algae, nostoc. This can be considered as a symbiotic association. This is the internal structure of Anthoceros. Okay. Okay, na?
Okay, ma'am.
From the lower epidermis of the thallus arise smoother walled unicellular rhizoids are present. It helps in fixing and absorption. Okay. So pegged rhizoids and scales are absent. Thallus is also divided of air chambers, air pores, and storage region. Okay, so this is the
Ma'am,
tell me.
ma'am, you said that it consists of upper epidermis and lower epidermis.
Yeah.
Ma'am, what is the meaning of upper and lower?
See here in the diagram, you observe that upper epidermis is there upside, lower epidermis is
Okay.
down. See here. At
Okay.
the bottom
Okay,
side,
ma'am.
the smooth rhizoids are there. This divided epidermis is dividing upper and lower.
Okay, ma'am.
In the center, this is the parenchyma cells.
Okay, ma'am.
ఇప్పుడు అర్థమైందా?
అర్థమైంది మ్యామ్, అర్థమైంది.
Okay. So next coming to the after the internal structure of thallus, we are coming to the reproduction. So Anthoceros reproduce through vegetative, asexual and sexual methods. In the life cycle, alternate generation of life cycle. So it shows two stages: gametophytic stage and vegetative and asexual reproduction. Okay, sporophytic stage, gametophytic stage. Two stages are there in the Markantia and Anthoceros also, Polytrichum also. Next lesson. Okay, so in the gametophytic stage, that is vegetative and sexual reproductions. In the sporophytic stage, that is asexual reproduction. Because of that, this is a haploid condition, this is a diploid condition. Remember this, gametophytic stage means vegetative and sexual. Okay, sporophytic stage means asexual reproduction. Okay, clear?
Clear, ma'am.
See here, we are coming to the gametophytic stage. Reproduction is two ways, that is vegetative reproduction and sexual reproduction. Vegetative reproduction. This occurs through fragmentation of the thallus, tuberous and gemma cups. Thallus, tuberous and gemma cups. Some rare gemma cups are formed in the Anthoceros, but Markantia is very important. Markantia and Anthoceros. Remember this, that through the vegetative reproduction, through the gemma, that vegetative reproduction in only we can observe in Markantia and Anthoceros. See here that in the thallus accidentally breaks down into small pieces. Each fragment develops into a plant. That is the fragmentation. Already you know this, very simple. Okay. By the tubers, in unfavorable conditions, some of the species develop tubers on the margins of the thallus. In the drought conditions, when the entire thallus degenerates in tubers remain alive with the thallus, in the commencement of favorable conditions, the tubers develop into the thallus. Okay, dumpalu antamu, tubers. So that is one of the vegetative reproduction. And the next is the gemma or gemma. Like Markantia, in some species, vegetative reproduction takes place through the gemma cups. So only two species, that is Anthoceros formosae and Anthoceros glandulosus.
Ma'am,
Tell me.
for tubers we can give examples like potato.
No. Tubers means like the tubers that formed in the Anthoceros thallus. So whenever favorable conditions are there, they can develop into the thallus. That is in Anthoceros we are observed tubers-like structures. That's why we are called the tubers. Vegetative reproduction of Anthoceros, not
Okay, ma'am.
okay.
Okay, ma'am.
So coming to the second one, sexual reproduction. Anthoceros gametophyte is a bisexual thallus and usually protandrous. What is the meaning of protandrous?
I don't know, ma'am.
Very simple. Protandrous
Ma'am,
means
really I don't know, ma'am.
male sexual organs, it may be develop than the before the female sexual organs. ఏదైతే మేల్ సెక్సువల్ ఆర్గన్స్ అనేవి ఫాస్ట్ గా డెవలప్ అవుతాయి అన్నమాట. దట్ ఈస్ ప్రొటాండ్రస్. ఓకే, సో దట్
Okay, then they are divided into parts. Ma'am, they are divided into parts, ma'am.
Tell me again.
You said no, ma'am. What is meaning of protandrous? They are divided into parts, ma'am.
No ma. So in the plant that is bisexual. Bisexual means male and female sexual organs. Okay,
Okay,
and
ma'am.
androecium and gynoecium is there in the plant. Male and female sexual organs. Okay.
Okay, ma'am.
So that male sexual organs can develop before than the female sexual organ. అంటే గైనీషియం కంటే ముందుగా ఆండ్రీషియం కనుక డెవలప్ అయితే దాన్ని ఏమంటామంటే ప్రొటాండ్రస్ అంటాం అనమాట. ఓకే.
Okay, ma'am.
So sexual organs are embedded in the thallus. Male reproductive structure is called, what is the male reproductive structure in the bryophytes?
Antheridium.
Antheridium. Female sexual organ is?
Archegonium.
Yeah. Okay, good. So see here, same in the, we missed in the structure of Markantia, but same. Okay. So what are the points lastly I explained in the Markantia Archegonia that you remember. Okay. So this is a Anthoceros antheridia structure. This is a club shaped structure. It is called antheridia. It is embedded in the thallus and antheridial chamber. This is we are called antheridial chamber. These are the antheridia. Okay. Each antheridium have two parts, stalk and stalk is multicellular and antheridium. Okay. Club shaped structure, antheridium. A mature antheridium is multilocular chambered with one cell thick jacket layer. See here, this is a jacket, Nana. This is a thick walled jacket layer and these are the cells, chamber, multilocular chamber. Each chamber of antheridium develops into biflagellate two male gametes or antherozoids. So each chamber can develop the two male gametes. Biflagellated two male gametes are developed from this antheridia. Are you understood antheridial structure?
Yes, ma'am.
Okay. Next slide. This is the development of antheridium. See here. But no need, Nana. The development is no need. Only structure is important. See here. Time waste. Okay. See here, same. This is a archegonia. This is female sexual organ of Anthoceros. Archegonium is a flask-shaped structure. So two parts are there, venter and neck. This is the egg cell. In the venter, that is egg cell is there, and neck canal cell is there. These are called neck canal cells. These are cover cells. Okay, lid cells are cover cells. This is a mature archegonium. So it has the two parts, that is the venter and the neck. Okay, the neck consists of 5 to 6 neck canal cells. So in the Markantia, how many neck canal cells are there? Already in the last class, I told. How many neck canal cells are there? What happened?
Ma'am, in my phone charging is completed, ma'am. In my phone charging is completed.
Okay. See here, 4 neck canal cells are there in the Markantia, but in the Anthoceros, the neck consists of 5 to 6 neck canal cells. Please remember this. This is very, very important number. Neck canal cells, 4 observed in the Markantia, but in the Anthoceros, 5 to 6 neck canal cells and 4 lid cells at the apex. The venter consists of a small venter canal cell and a large egg cell. See here. This is the venter canal cell. This is the egg cell. These are the neck canal cells. Clear? So after matures, archegonium lid cells separate out, neck canal and venter canal cells dissolve and release a mucilaginous chemical substance. From that canal, male gametes enter into the archegonium and fused with the egg cell form into zygote. This is the about the archegonia. Are you understood?
Yes, ma'am.
Okay. This is a flask-shaped structure. It has two parts, venter and neck. The neck consists of 5 to 6 neck canal cells and 4 lid cells. It's common. Lid cells are common, but neck canal cells are 4 observed in the Markantia, 5 to 6 are observed in the Anthoceros. The venter consists of small venter canal cell and large egg cell. These two are dissolved. It forms the chemical substance. Mucilaginous chemical substance is formed after the dissolution of neck canal cells and venter canal cells. From this canal, the male gametes are enter into the archegonium and fused with egg cell, form into zygote at the fertilization time. Okay? Okay.
Okay, ma'am.
So next, this is a fertilization. As the archegonium matures, the lead cells are separate out, and these neck canal cells and venter canal cells dissolve a mucilaginous chemical substance. So that these are attracted. Antherozoids are attracted towards this mucilage. So enter into the archegonium and swim in the water to reach the egg, and fertilization takes place. This fertilization is called chemotactic fertilization. Because of that mucilaginous chemical substance attracted to the antherozoids. That's why we are called chemotactic fertilization. Okay, this is fertilization. Next one, sporophyte stage. Okay, so after that, when the formation of zygote, that is the diploid stage, the zygote develop into a sporophyte embryo, and after that embryo can develop the sporophyte stage. Okay, gametophytic stage is completed. Are you understood? Okay.
Yes, ma'am.
Okay, ma'am.
So coming to the sporophytic stage, development of sporophyte structure, and then let's see here, sporophytic stage. The zygote develops cell wall, undergoes division, and develops into a sporophyte. This shows diploid phase. Remember this, gametophyte is haploid stage, and the sporophyte is a diploid stage. Okay. So this sporophyte of Anthoceros develops on the gametophyte with elongated, linear, green, horn-like structure. So called these are hornworts. Okay. Sporophyte of Anthoceros consists of 3 parts: foot, middle meristematic tissue, and capsule. So what you observed from the Markantia and Anthoceros? What is the difference, Nana? Please tell me. Foot, middle meristematic tissue, capsule. These parts are present in the Anthoceros. What parts observed in the Markantia?
Ma'am, in Markantia, foot, seat, and capsule. Foot, seat, and capsule.
Yeah, very good. Foot, seta, and capsule is observed in the Markantia, but in the Anthoceros, the seta is absent. Okay, seta is absent. So instead of seta, here middle meristematic tissue is present.
Okay, ma'am.
The capsule is covered by a tubular involucre, which is derived from the gametophyte. Okay, so the capsule is covered by that protective involucre, which is derived from the gametophyte. See here. So this is the LS of sporophyte. The sporophyte of Anthoceros consists of 3 parts: foot, middle meristematic tissue, capsule. Okay. See here. This is a foot. Okay, you observe, clearly observe that foot. Matured sporophyte shows a massive bulbous foot, which is embedded in the gametophyte. It helps in the absorption of water and minerals from the gametophyte. This partially dependent on the gametophyte. So that's why that food is observed from the gametophyte, that the minerals and water. Okay, that is the food function. Okay, so next, this is a intercalary meristematic zone. That is the meristematic tissue. Okay, there is no stalk-like structure between the foot and capsule. That is, the seta is absent in the Anthoceros. So that foot and capsule in the sporophyte of Anthoceros. So foot and capsule in between, that seta is absent. Here, intercalary meristematic region is there. Instead of seta, intercalary meristematic tissue is present. This is very, very important. 99% in the we have to expect this question. Okay, so in the Anthoceros, seta is absent. calorie meristematic region is present, or meristematic tissue is present in between the foot and capsule. Remember this, very, very important. Okay. In the Anthoceros sporophyte, we are observed that foot and intercalary meristematic tissue and capsule. Okay, all are you understood? Okay, no.
Yes,
This region
ma'am.
shows divisions continuously and continuous growth, the growth of the capsule. See here, that region, whereas the meristematic region is there, that undergoes divisions continuously and continues the growth of the capsule. This meristematic tissue remains for a longer time and helps in the growth of the capsule even after the spores start released. Because of this, the sporophyte growth is considered indefinite, able to develop for functional longer period and release spores continuously. This is a capsule, okay? So that spores are released continuously. This sporophyte growth is considered indefinite. So that's why this is able to develop for functional longer period and release the spores continuously. So see here, this is a spore. This is a elater. Pseudo-elaters are present in the Anthoceros. Okay, so next one is the capsule. So this capsule is a vertically growing, lean, elongated, horn-like structure, measures 3 to 10 centimeters in length and is green in color. The central part of the capsule consists of 16 vertical rows. See here, vertical rows in the middle of the center of the sporophyte. Okay, this totally we are called capsule. Okay, this is the meristematic tissue and this is the bulbous foot. And this is the capsule. The central part of the capsule consists of 16 vertical rows of sterile cell constituting of columella. This we are called columella, the center that vertical rows. Okay, and encircled by archisporium. Outer wall layers are present. Columella helps in conduction of water and provides mechanical support. Okay, so this columella, center of this sporophyte, it's 16 vertical rows are there. This is helps in conduction of water and provides mechanical support. It gives the mechanical support and conduction of water. Okay, that is the columella function. See here, the capsule wall is 4 to 5 cell thickness. The outer cells are differentiated into epidermis with stomata here and there. See here, and sometimes this stomata are also present in the outer layer. Like the evolved plants, the stomata have 2 god cells. Smaller 8 chambers are present below the stomata. Next to the epidermis, the inner layer cells have chloroplasts. So the sporophyte can synthesize its food and depend on the gametophyte for water and minerals only. Okay, this sporophyta outer epidermis and before that, so below the epidermis, some layers are there. So these having that inner layers have the chloroplast. So what happened? That chloroplast can synthesize the own food. So only they depend on the gametophyte for the water and minerals only. Okay, are you understood? సో డిపెండెంట్ అన్నాను కదా, మళ్ళీ అదేంటి ఓన్ ఫుడ్ సింథసైజ్ అవుతుంది అంటే క్లోరోఫిల్స్ ఉంటాయి కాబట్టి ఫుడ్ ఓన్ గా ప్రిపేర్ చేసుకున్నప్పటికీ వాటర్ అండ్ మినరల్స్ మాత్రం డిపెండెంట్ గా ఉంటుంది ఫ్రమ్ ది గ్యామెటోఫైట్. సో అదే ఎవరు అబ్జర్వ్ చేస్తారు? ఫుట్ అబ్జర్వ్ ది వాటర్ అండ్ మినరల్స్ ఫ్రమ్ ది గ్యామెటోఫైట్. That's why the sporophyte of Anthoceros is partially parasitic. That's why we are called partially parasitic. But Markantia sporophyte is parasitic. We are completely parasitic. But here Anthoceros sporophyte is partially parasitic, partially dependent on the gametophyte. See here that archisporiums are present below the wall layers of the capsule. This we are called archisporium. Okay. So this gradually increases up to 2 to 4 cells. These archisporium consists of spore mother cells and pseudo-elater mother cells in an alternate manner. Okay, ఇక్కడ మనకి ఏవైతే మిడిల్ సెంటర్ లో ఉన్నాయో దట్ we are called pseudo-elaters. Okay, so reduction division takes place in the spore mother cell and develops spore tetrads. So then the cells are going to meiosis. So that's why haploid spores are formed as tetrads. First develops spore tetrads. 4 ఒకే దగ్గర ఫామ్ అవుతాయి కాబట్టి we are called as tetrads. And pseudo-elater mother cells develop into pseudo-elaters, which differ from the elaters of Markantia in the absence of spiral thickenings. Okay, So what is the special of elaters and pseudo elaters? So elaters having the spiral thickenings, but pseudo elaters absence of spiral thickenings. Remaining function same, but only spiral thickenings are absent in the pseudo elaters. So at the apex of the capsule, the spores and the pseudo elaters are free, and the columella helps in the breaking of the capsule wall. So then after the development of spores, these spores are small, spherical, black in color and have light. This is the spore dispersal. When these spore pseudoelaters absorb the moisture, the matured capsule is brown or black in color. By losing water, and it gets dried up, and the capsule splits or dices longitudinally. When the released spores fall on suitable substratum, they germinate and develop into the gametophyte. Same. Okay. So when the spores fallen on the suitable substratum, so they can develop or germinate to the into the gametophyte. That is the spores dispersion. This is a life cycle of Anthoceros. See here. So some vegetative and sexual reproduction coming under the haploid. So that is a gametophytic stage, and after that asexual reproduction, that means sporophytic development, that is sporophytic stage, that is the asexual reproduction. This is the sexual reproduction. So off is there, alternate generation of cycle, life cycle. Okay. So then the spores are developed and germinate as gametophytes. So same like the after the gametophyte development, sexual organs are developed on the gametophyte, antheridium and archegonium. So then after the development of the two sexual organs, egg cell and antherozoids are fused, and at the fertilization time, same zygote is formed. After the zygote development, they can develop as the sporophyte. Okay, sporophyte and gametophyte coming alternatively. So that's why we are called that life cycle is alternate generation of life cycle. Markantialo ante, Anthoceros lo kuda ante. This is about the Anthoceros type study. What are you understood?
Yes, ma'am.
Okay, na? If you have any doubts, please ask me from the Anthoceros. No doubts?
Ma'am,
Tell me.
you said that intercalary meristematic region, it contains three types, no, ma'am.
Intercalary meristem is there in the sporophytic structure.
Meristematic region. Tissue, ma'am. Once show the slide, ma'am, that I'll ask that question.
Oh. Okay. Instead of theta, intercalary meristematic tissue is present. This region undergoes divisions continuously and continues the growth of the capsule. So that's why the capsule grows from the base onwards.
Okay, ma'am.
This meristematic tissue remains functional for a longer time and helps in the growth of the capsule even after the spores start being released. And spores release అయ్యేంత వరకు అది డెవలప్ అవుతూనే ఉంటుందన్నమాట. Okay, so that's why the growth is considered indefinite, able to develop a longer period and release spores continuously. That is
this Anthoceropsida is there, no, ma'am. I bought this Anthoceropsida in exam. They will give question, ma'am.
I didn't understand, ma.
You explained now just now about Anthoceropsida, no, ma'am.
Anthoceros type study.
That about Anthoceropsida in CPGET exam, they will give compulsory question. I'm asking.
From the BRYOPHYTA, 1 or 2 questions compulsory, no? That may be Markantia or Anthoceros or Polytrichum. We don't know.
Okay, ma'am.
So these are the 3 type studies in your syllabus. 3 type studies are there.
Okay.
Okay.
From the Bryophyta, Markantia and Anthoceros and Polytrichum.
Okay, ma'am.
Tomorrow onwards, I will explain that Polytrichum that belongs to Bryopsida. Hepatic
Okay,
apsida, Anthoceratopsida, Bryopsida.
Ma'am, tomorrow also there are class?
Monday, I think.
Okay, ma'am.
Okay.
Okay, madam. Okay, thank you very much. If you have any doubt, please ask our respected madam.
Sir,
Okay.
I already asked madam.
Okay, otherwise, I will going to wind up the session. Madam has clearly explained about the internal structure and external morphology and also reproduction of Anthoceros, the beautiful structure of the Archegonian Antheridia. My dear friends, Archegonia is one of the most important structure. Why? Because any competitive exam, Archegonia contains how many neck canal cells or ventral cell or the internal structure of the thallus. Also most important, my dear friends, even though the reproduction is most important. So I hope class is very well. Tomorrow, same time, we will going to meet.
Sir,
Madam.
sir, not madam. I'm talking Srinaina.